Hi, my name is Robin Bond, and I am a scientist. If you ask me what sort of scientist I am, I would probably say I'm a biogeochemist. Now, that may not be a sort of scientist you've ever heard of before. Basically, I'm the sort of scientist who looks at a lot of different fields of study, and specifically, I like to look at how microbes, like these bacteria, affect the chemistry of the environment that they live in. What sort of environments? Well, I've done some work out on a dry lake bed that's super salty. I've looked at microbes out in the middle of the Pacific Ocean. And most recently, I've been looking at salty lakes. So basically, you can just say, I'm salty. One of my favorite things to look at is chemical kinetics, which is basically how fast a chemical process occurs. Now, you probably know from observation that some things happen faster than others. For example, in this video here, I'm doing a reaction which is very similar to something you might have done yourself. I'm putting baking soda into an acid. And you can see that reaction happens pretty fast. Another reaction that you might see happening is that if you get a nice new car, it might eventually rust because iron does that. And that is not a fast reaction, which is really great because if you had a car that started rusting as fast as the baking soda reacted, your car would be falling apart a week after you bought it. And that would kind of suck. When we study kinetics, what we like to do is we like to see how the quantities of things change over time. So, for example, here are two things, and we're going to graph how much of it is left over time. And you can see that over time, both the red and the green thing are decreasing. But if you take those points that you measure and you actually draw a line through them, you can see a trend, and you can see that the green one is decreasing much faster than the red one. And we can actually use this to create a predictive model. So if you've taken algebra, you probably know that if you have some points, you can calculate the equation for a line, and that helps you predict other points on the line. That's a very simple model. In real life, things aren't always lines. So here's an example of some things that I made predictive models of. I looked at how they changed over time, and I fit them to an equation. And you could see that they're not a line, but we can find a predictive model nonetheless. So let's say that you have this model. You've taken these data points, and you put a line through them, and it's pretty much flat. So if you see this model, does that mean nothing is happening in your system? I want you to think about that for a bit. And let's look at some real life situations. Okay? So you probably know that plants produce oxygen. And let's say this particular plant is producing 10 milliliters of oxygen per hour. So after 1 hour, it's gone up 10 milliliters. So there's our point. An hour later, it's gone up another 10. There's our point. An hour later, it's gone up another 10. There's our point. And now you can see we can draw a line through all of these and predict how much oxygen there will be at any point in the future. Here's another thing that might happen. We know that animals breathe oxygen. So here's this little mouse, and let's say that this mouse uses up 10 milliliters of oxygen per hour. So 1 hour later, we'll be down 10 milliliters. Another hour later, we'll be down another 10. Another hour later, we'll be down another 10. And let's draw a line through that, make a predictive model. Oh, look, this predictive model shows the mouse is going to run out of oxygen in a little over 3 hours. Now let's take our plant and our mouse and put them in the same room. The plant creates 10 milliliters of oxygen every hour, and the mouse uses up 10 milliliters of oxygen every hour. So after 1 hour, the plant has made 10 mils of oxygen. The mouse has used 10 mils of oxygen, and look where we are. An hour after that, we've got 10 more milliliters of oxygen from the plant, 10 fewer from the mouse. That's where we are. And we could go on and on, but let's just draw a predictive model through this. This is what's happening to the amount of oxygen in the room. The amount is not changing. Does that mean nothing is happening? No, things are happening. It's just that the processes are working against each other. And what we call the net change, the sum of all the changes, might be zero, but there are still things changing. So that's really important to understand that these processes can work against each other. But what if the rates were different? We can also look at these processes when they are at different rates, which is more common, right? So here the plant is going to make more oxygen than the mouse uses up, more oxygen than the mouse uses up, and we can see that over time, sure, it's increasing. It's not increasing necessarily as fast as it might have in the past because the mouse is using some of it. And if the mouse used more than the plant made, then the amount of oxygen would decrease and eventually the mouse would run out of oxygen again. So it's really important to understand the speeds of these reactions that work against each other. I've showed you this example with oxygen because that's something that people see every day and they understand it. I like to look at a different chemical. I like to look at hydrogen peroxide. Now you may think that hydrogen peroxide is only found in the pharmacy or in those brown bottles in your medicine cabinet, but that's not true. Hydrogen peroxide is all around you, and not only that, it's in you. You see, you take oxygen and you turn it into water, and your body does that because it gets energy. That reaction is actually one of the most important things that makes energy for you to live on. Now, of course, plants do the opposite. They take water and they turn it back into oxygen. And because it's going backwards, it doesn't make energy for plants. It actually requires energy to go backwards. And plants get that energy from the sun. The thing about this reaction of oxygen turning into water is that it's actually a multi-step, very complicated reaction. And as the steps go on, we make what we call intermediates, things that are formed between oxygen and water. The first one we call the superoxide radical. It's oxygen with an extra electron. The second one is hydrogen peroxide, H2O2. And the third one is what we call the hydroxyl radical, OH. Those three intermediates we sometimes refer to as reactive oxygen species. And they're really important in our body and in the environment. So let's look a little more closely at why they're important for living things. So these are called reactive oxygen species because they react really easily. And they react with things in your body or in other organisms. For example, they can damage proteins, which is not great because a lot of things that your body does, it does with proteins. They can damage fats, which is terrible because your cell membranes are made up of fats. And you kind of need your cell membranes to hold your cells together. They can damage DNA. And that's really bad because without DNA, your body can't repair itself. So you can probably understand why these things are so bad. They can kill your cells. Or even if the cells aren't dead, they can cause cancer. Yeah, that's right. These are those famed free radicals. So when you hear people tell you to take antioxidants to avoid cancer, they're basically trying to stop these reactive oxygen species from working in your cells. All those bad things. And yet, living beings use reactive oxygen species because they're so helpful. Superoxide and hydrogen peroxide are used in your body. For example, remember I said they kill cells? Well, your body uses superoxide to kill cells. Not your cells, but pathogens. Anything that could make you sick. Your immune system uses phagocytes. And they find those intruders and they hit it with superoxide. And that kills it because superoxide kills cells. Hydrogen peroxide is great at breaking down fats and you don't want that near your cell membranes. But if you eat some olive oil or something like that, your body needs to break it down in order to use it. And hydrogen peroxide can be used for that. And there are some organisms We aren't some of those organisms, but there's some organisms that use reactive oxygen species to absorb nutrients, like iron. Iron is a really important nutrient for microbes in the ocean. And you probably know that if your body needs iron, you can't just eat a nail. It has to be in a form that your body can absorb. And reactive oxygen species can take iron, that's like a nail, and turn it into something that your body can use. So here we come to the cool thing about hydrogen peroxide. We have many organisms that make hydrogen peroxide because it's useful. That is going to overall increase the hydrogen peroxide in a system, whether it's inside your body or outside in the environment. At the same time, we have many organisms destroying hydrogen peroxide because it's harmful, and that decreases the amount of hydrogen peroxide in the environment. Once again, we have these two processes that are working against each other, and it would be really nice to know overall what happens to the level of hydrogen peroxide in a system, but also, can we measure both processes at the same time? I have done that in many places on Earth, as I've shown you before, but I'd like to talk to you about a really cool place where this is important because it affects astrobiology. You see, some of us are really fascinated with whether or not life exists elsewhere in the universe, and specifically in our own solar system. And the way that we study astrobiology without being able to leave the planet is to look at what life on Earth does and try and apply it in other environments. So let me give you an example. We talked about oxygen cycling on Earth. We have oxygen. Through respiration, we turn that into water. That's why if you breathe on a cold piece of glass, it fogs up. That water comes from your breath, right? That gives us energy. And then we can't stop there, because if all we did was turn oxygen into water, soon there would be no oxygen left on Earth. There would be a lot more water. So somehow, to keep the cycle going, the water has to be turned back into oxygen. And plants do that, as we talked about before. That needs energy that comes from the sun. But what if you were on a place where there wasn't a lot of sunlight, like Europa? Europa is one of the moons of Jupiter, and it's one that we're really fascinated with because it has a lot of water. It has more water than Earth, and that's great for the possibility of life. Europa is really far from the sun, so far that photosynthesis probably wouldn't work. And also, it's got a whole bunch of ice between the ocean and the outside. So even if the light could get there, it couldn't get through the ice to where it really needs to be. But Europa has something really weird going on with it. It's really close to Jupiter, and Jupiter has a huge magnetic field, 20,000 times stronger than Earth's. And that magnetic field is always sending out these little charged particles through space, and those charged particles hit the ice on Europa's surface, and that turns it into hydrogen peroxide. So there's actually an awful lot of hydrogen peroxide on Europa. If that hydrogen peroxide can get down out of the ice into the ocean where it can move around and bump into things, then it could possibly turn into oxygen. And so if you look at Europa, we kind of hope that there are things living on Europa that would breathe in oxygen and put water out, and that would give them the energy to live. But we also need something that's going to bring the water back to oxygen, just like there is on Earth. That's not going to be photosynthesis. What it's going to be is the energy input by Jupiter's magnetic field that turns water to hydrogen peroxide, which then turns back into oxygen. And people have done calculations, they've done computer models, and they have figured out that there could be enough oxygen put into Europa's ocean to support millions of tons of fish. So that's great. But what does this have to do with kinetics that we were talking about before? Well, let's look at what happens when the hydrogen peroxide goes into the ocean. We have hydrogen peroxide coming into the ocean from the ice. And remember, it's a reactive oxygen species. It's really reactive. It is going to react with something. And there are two things it's most likely to react with. It can react with itself. That turns it into oxygen. Or in that ocean, people think that there's a lot of undersea volcanic activity. It can react with metals that come up from those undersea volcanoes. So those are the two possibilities. If it turns into oxygen, well, oxygen can stay in water to a certain extent. For example, fish breathe oxygen in water. They need oxygen just like us. So some oxygen can stay in the ocean. But there's a limit to how much oxygen can do that. If you get a lot of oxygen, what ends up happening is it forms bubbles and it comes out of solution and it bubbles up to the top of the water. Now, if you have metals coming up from the undersea volcanoes, and if that's what's reacting with the hydrogen peroxide essentially makes things that are like rust. If it reacts with iron, it's going to make something that is almost exactly like rust. Or it could react with other metals to make similar things. But you know what rust is like. Rust does not dissolve. So the rust gets formed, and it settles down to the ocean floor. So you could say, which of these things happen? Does it turn into oxygen? Does it turn into rust? Well, both. So the real question is, which does it do faster? I want you to stop and I want you to think about what would happen if each of these was faster. What would happen if you formed rust faster than oxygen? And what would happen if you formed oxygen faster than rust? So if you form rust faster, all that hydrogen peroxide that reacted with the metals is no longer available to turn into oxygen. And the rust, as I said, settles down to the ocean floor. You're going to get a layer of rust on the seafloor. And this is not very useful to any animals that want to use it to live. So that is something that is not good for life on Europa. If oxygen formation is faster, then before hydrogen peroxide can react with the metals, it is going to turn into oxygen. Now, dissolved oxygen can still react with things like metals, but if oxygen is formed fast enough, there's going to be oxygen coming out of solution and turning into bubbles. And what you end up with is oxygen bubbles that are going to rise to the surface and get trapped under the ice crust. Once it's no longer in the ocean, it can't react much with anything in the ocean. But this is a much better scenario for organisms that might want to use oxygen for life. So life can exist on Europa if you have the right chemical reactions happening, and we think that they do happen, but they also have to happen at the right speed. I hope you learned a little bit about chemistry and about life on other planets. Thanks for listening.